Hi everyone, my seminar topic is muscle development and maintenance in insects. We are already studied about the muscle development in developmental biology. We know that the muscle fibers are formed by the fusion of the myoblast, and this process is also similar in the case of insects also. The muscle development begins with the specification of the myoblast from the mesodermal cells during the embryonic period. And when the muscle fibers are formed and that embryonic muscle pattern remains constant during the later stages of the insect development. The fibers is grow along with the organism by the increase in the fiber volume, and it is not by the nuclear division or the myoblast fusion. There would be a major differences between the muscle development in the holometabolous and the hemimetabolous insects because of the pupal life in the holometabolous insects. During the embryonic myogenesis in the holometabolous insects, a large fraction of the myoblast in each segment is put aside as a separate nest, or they are arranged within the imaginary disc, and there should be an increase in number but do not differentiate. At metamorphosis, most of the larval muscles are destroyed, but some of the larval muscles are persist, and these muscles serve as a template for the dorso-longitudinal flight muscles, and they also continue their function during the eclosion. And most of the other muscles that present in the pupa are de novo in origin, and that mainly include the dorsoventral flight muscles. They are formed by the division and fusion of the myoblast also. Muscles often continue their development in the early days of adult life also, and this was reflected by the insect's inability to undertake their extended flight during that period. And that this period, that is after the eclosion, during which an adult insect's inability to fly is found, and that period is known as the teneral period. The flight muscles may regress in adult that have completed their flight period, and some of the muscles is also regressed or completely disappeared. Example, the hatching muscles. For hatching, the muscles associated with the hatching is degenerated or regressed after completing that process. In the case of holometabolous insects, some of the larval muscles may pass to unchanged into adult, and some other muscles are reconstructed. Some of the larval muscles may be destroyed and not replaced, and new muscles are also formed, such as the flight muscles and leg muscles are also formed from the imaginary discs. And next we're going to discuss about the muscle development in the hemimetabolous insects. In hemimetabolous insects, increase in muscle size is found in the larval development and also some period after the eclosions also. And the growth during the larval period is occur because of the increase in the number of fibers. And the growth after eclosion is found by the increase in the size of fibers. And this is a major difference between the muscle development during two periods, and these difference point out the fact that the new attachment to the cuticle are normally formed at that moment. The difference in the number of fibers between early stage larvae and adult is relatively small in the muscles, which perform same function throughout the life of the insects, but more marked in the case of flight muscles. The flight muscles are also only found in the adults. So there should be differences in the number of fibers between the larvae and the adult associated with that flight muscles. And the increase in the fiber size also involved the increase in the myofibril, mitochondria, sarcoplasmic reticulum is also. The relative rate of increase of these components varies from muscles to muscles also. In the case of grasshopper, Cystocerca nitens, all the components of the pterygococcal muscles increase in proportion with the larval development. But in the case of metathoracic dorsal longitudinal muscles, the muscle volume occupied by the myofibrils doubles during the last three stages. In both of the cases, the proportion of muscle volume occupied by the mitochondria increases during the first 2 weeks of the adult life. In the case of Locusta, The fiber growth in young adults involve an increase in the size of myofibrils. And in the dorsal longitudinal muscles, a doubling of number of filaments is also found similar to the grasshopper. And these anatomical changes are mainly brought about by the enzyme activity. Some of the enzyme activity will be increased during that period, the period after the eclosion, when the flight metabolism is active. And this feature is shown in this graph, the enzyme activity at the different stages of the insect development. Here the zero indicate the eclosion, and 7 days after eclosion, most of the enzymes reaches a plateau level. Their concentration is increased, and that is responsible for the ability of the insect's flight. And in that time, lactate dehydrogenase is degenerated. So, the activity of enzymes involved in the flight metabolism increases through the final stages of the muscle development, and that tending to reach a plateau level in about 7 days after the eclosion. At that time, the activity of the lactate dehydrogenase declines, and that is responsible for the flight ability in the insects. The post-eclosion growth of flight muscles of holometabolous insects. During the time of eclosion, when the cuticle expands, the flight muscles of Cycloraphus flies rapidly increase in length by 25% or more. Cycloraphus flies. This is the picture of Cycloraphus flies. It's belong to the order suborder Cyclorapha. In the Cycloraphus flies, at the time of eclosion, flight muscles is found to be increased in length, and this is occurred by the increase in the length of the sarcomeres and their filaments, and is not because of the change in the number of filaments. And this increase in the flight muscle volume is also found in the Hymenoptera insects also. In the case of tissy fly Glucina, post-eclosion changes is similar to the Orthoptera, but the flight muscle maturation is depends upon the insects that feed on the blood meals. The mass of muscles is increases with the increase in the size, and within each myofibril, the number of thick filaments more than doubles, and also the mitochondria increased in size. At the same time, the wing beat frequency also increases, and this is mainly due to the changes in the cuticle and also the increased muscle stiffness. And these comparable changes is also found in other flies also. In the case of honeybee apis, the mitochondria volume is increases 20-fold during the first 3 weeks of the adult life. And this is not found in the case of Drosophila, where the flies can fly within a few hours after the eclosion. So these changes is not found in the Drosophila. In the case of the Lepidoptera, no post-eclosion changes is occur. The flight muscles are fully functional after the wing expansion. Next, we are going to discuss about the regressive changes in the flight muscles. In the case of termites and the ants, the flight muscles of the reproductive cars are regressed after the nuptial flight, and this flight muscle degeneration is also found in the crickets also. And this regressive changes associated with the muscles is also found in other group of insects also. That is bark beetle. This is a picture of bark beetle. In the case of bark beetle, the degeneration is followed after a period of reproduction, and the redevelopment of the muscles is occurred during the further flight. In the case of Colorado potato beetle, this temporary regression of the flight muscles is mainly associated with the reproductive diapause. And this is the picture of the Colorado potato beetle. And this muscle degeneration is correlated with the increase in the juvenile hormone titer, and that regulate the reproductive development. In the case of most of the insects, this muscle histolysis is correlated with the increase in the juvenile hormone concentration. But in the case of Leptinotarsa, the insects enter diapause and the flight muscles degenerate when the juvenile hormone titer is low, and they regenerate when it rises. So In the case of leptinotarsa, the flight muscle degeneration is occur when the juvenile hormone titer is low, and these flight muscles regenerate when the juvenile hormone concentration rises. And in the case of aphid acrothocyphon and in other insects, the breakdown of these indirect flight muscles is a genetically programmed event. And this regression of the flight muscles is also found in other group of old species also. And this is the picture of acrothocyphon aphid. Next, we are going to discuss about the muscles associated with the hatching, eclosion and the molting. The muscles associated with this hatching are mainly found in the grasshoppers and the crickets. In the case of newly hatched grasshopper larvae, there should be a pair of ampullae in the neck membrane that are mainly used to escape from the eggshell, and also their use is no longer when the insect reaches the surface, so that their associated retracted muscles get degenerated after the insect reaches the surface. The muscles associated with the molting are also active at this time, but they do not degenerate after hatching. In the Rhodnius is an assassin bug. The muscles that develop at each molt are also present when the insect hatches, but regress shortly afterwards. Muscles associated with the molting is also found in the hemimetabolous insects also, and their function is to facilitate the ecdysis and the expansion of the cuticle at molting by increasing the hydraulic pressure of the hemolymph. In the case of grasshoppers and other insects, some of these muscles are attached to the membranous regions of the cuticle, and they may prevent excessive ballooning of these areas, so that the greatest force are extended on the presumptive sclerates, ensuring that should ensure maximum expansions. In the case of grasshoppers, there should be accessory muscles in the neck, thorax, and the abdomen, and which regress after the molting. In the blood-sucking bug Rhodnius, some ventral intersegmental muscles in the abdomen are fully differentiated only when the insects molt. And within a few days of molting, they regress and lose their contractile function, and they redevelop in the readiness of the nest molt when the insect has a blood meal. So in the case of Rhodnius, most of the ventral intersegmental muscles in the abdomen are differentiated only when the insect molt, and within a few days of molting, that they will get regressed and lose their contractile function, and these muscles get regenerated during the nest molt. Among the holometabolous insects, muscle that function at the time of eclosion is also only found in the diphtheria and the lepidoptera. In the case of blowfly sarcophaga and also in the Drosophila, and these special muscles in the abdomen that are mainly involved in the escape from the puporium and the subsequent cuticle expansion. And these muscles are known as the persistent larval muscles, and they are extended from the front of one presumptive sclerate to the front of the nest. In the newly emerged adults, of contraction of these muscles causes the sclerate to buckle because they are not yet hardened and the abdominal cavity is reduced in the volume. It is these movements that hydrostatically cause the erosion of the pitilinium. Pitilinium is an frontal organ that was found on the head of the sarcophaga and the Drosophila, and that is required for the breakdown of the puporium and also the eclosion. The muscles associated with that is also breakdown when the cuticle is expanded and the hardened. The first stage of the breakdown is these muscles in the lepidoptera is mainly triggered by the falling concentration of the molting hormone. So in the Lepidoptera, and this muscle breakdown is occur by the when molting hormone concentration fall down, and it is occur before eclosion when the but it is delayed if the motor neuron controlling the muscles are active. In the case of tobacco hornworm moth Manduca, The absence of molting hormone contribute for the complete disappearance of the muscles in the days after eclosion. But in the case of silk moth and the ray, eclosion hormone provide a signal for the degeneration. So that will be the major difference. That is, in the Lepidoptera, muscle breakdown is contributed by the decrease in the concentration of molting hormone. But in the case of silk moth, and this eclosion hormone provide a signal for the, that is, they with increase in the concentration of molting or the eclosion hormone, and these muscles associated with that get degenerated. In the flesh fly sarcophaga, eclosion hormones start a slow degeneration, but some other factor triggers the rapid degeneration of the thoracic eclosion muscles also. And the effect of these hormones is to switch on the genetically programmed cell death. And these hormones are mainly contributed for the genetically programmed cell death, that is apoptosis. Next, we're going to discuss about the effect of some activity on the muscle development. Some of the examples are known in which the muscle size is affected by the activity. In the Glossina, the flight following the eclosion result in the rapid increase in the mass of the flight muscles, and this increase occur more slowly in the absence of induced activity. So the flight is contributed for the increase in the flight mass of the flight muscles in the case of Glossina. But when this activity is absent, that growth is not occur, or that growth is occur very slowly. And the hardness of the foot also affect the size of the mandibular adductor muscles in the caterpillars of the moth Pseudalacia. And this, so in the case of moth Pseudalacia, uh, hardness of the foot affect the size of the mandibular adductor muscles. When the insects feeding on the harder food, that will develop larger heads and the larger muscle than those eating the soft food. And similar changes is also occur in the grasshoppers also. The regression of unused muscles can also occur, but it is not a consequence of the lack of use. The regression of flight muscles in numerous insects follows loss of the wings and is probably controlled by an increase in juvenile hormone titer in the hemolymph. The grasshoppers can autotomize their hind legs, that is, separate their hind legs, a break appearing between the trochanter and the femur, and that may contribute for the degeneration of the thoracic muscles also. So in this video, we are discussed about the muscle development and maintains in the insects, especially in the holometabolous and the hemimetabolous insects, and also the regressive changes stored in the flight muscles, and also the muscles associated with the molting, hatching, and the eclosion, and also the activity of different activity on the muscle development. Thank you.